I'd like to start off today by welcoming everybody to the National PKU Home Phe Alliance webinar. Absent a cure, one of the major challenges faced by PKU patients is monitoring their blood phenylalanine or blood Phe levels and making real-time changes in their treatment in response to those Phe levels. Today, first-line therapy for PKU is a highly restrictive diet where keeping blood Phe levels in the safe zone requires very restricted protein intake. As new therapies become available, some PKUers are now facing the new challenge of blood Phe levels that are too low, increasing the need for frequent and real-time blood Phe monitoring. Recognizing this problem, the National PKU Alliance has made the development of a Home Phe monitor that can give fast, accurate, and cost-effective results at home a high priority. In 2013, the National PKU Alliance partnered with Incentive Incorporated to challenge the scientific and technical communities around the world to develop a simple and cost-effective Home Phe monitor that would provide PKUers with real-time blood Phe analysis. Together, the National PKU Alliance, Incentive Incorporated, and the Cundill Foundation raised a total of $776,000 to fund the Home PKU project. The National PKU Alliance and its Scientific Advisory Board were tasked with the financial and scientific oversight through the 3 phases of this project. As with the development of any new technology, some prototypes did fall by the wayside, while others continued to progress. Those that were showing promise were tested in a joint project with Emory University in 2015. Then, in the spring of 2019, the National PKU Alliance and its Scientific Advisory Board held a meeting in Seattle, Washington, where the 3 most advanced developers gave live demonstrations of their prototypes using blood drawn on site from PKU and non-PKU volunteers. While the results compared very favorably to the blood cards set up at the same time, the demonstrations also revealed the need for some additional development. In today's webinar, the National PKU Alliance is pleased to introduce 3 companies that are on track to launch Home Phe monitors in the near future. Each company will have about 10 minutes to present their technology and their commercial development timelines. At the end of the 3 presentations, we'll have time for some questions. To keep the presentations on schedule, please hold your questions until the end. Questions can be asked through the chat function of the GoToMeeting software. The chat control button can be found in the upper right-hand corner of the screen. The speakers today are Michael Boyce-Aquino from Aptatek Biosciences, Peter Warto from Egoo Health, Shima Dalarirad and Robert Harper from In Vitro Diagnostic Solutions. Welcome to the National PKU Alliance Home Phe Monitor webinar. And Michael, let's begin with you.
Thank you, Lex, and thank you to the NPKUA for the opportunity to speak to everybody today about Aptatek's home monitor for PKU. Next slide, please. So I'm Mike Boyce-Aquino, as Lex mentioned, CEO of the company. Tila Wargal, our Chief Medical Officer, Milan Stoljanovic, our Chief Scientific Officer, are co-founders and co-inventors of the technology that I'll be talking about today, and they're at Columbia University in New York. Next slide. So I wanted to preface the discussion with a couple of needs really for home monitoring that are important in thinking about these devices. We want a test that's quantitative. In other words, giving you results that are analogous to what you're getting from your current testing in the clinic. So we also want, of course, that accuracy and precision that's equivalent to that clinical lab data so that both you and your clinicians can trust that data in managing in disease management, and really have that validated through both regulatory process of FDA clinical trials and sort of peer testing within the clinical community in the PKU field. And make it easy for the user, of course, is really important, especially for home monitoring. So minimal finger stick of blood is where we targeted 5 microliters of blood, automated as much as possible. So the number of steps where it gets too complicated or things can go wrong, we want to minimize those, and make it connected. So the results that you get aren't just a number that you might see, but actually, or like a pregnancy test, it's not just plus minus, but ideally giving you a result that you can store, compare over time, and share with your clinician as well. Next slide. So, and one of the other challenges we want to think about with PKU, of course, is that phenylalanine, which is the target molecule here, is a very, very small molecule, 900 Daltons, meaning it's many thousands of times smaller than our unfortunate coronavirus right now. So it's hard to detect with standard clinical approaches to point of care or home testing, which would be antibody-based tests most typically. It's too small for that antibody to really accurately stick to and detect phenylalanine. So we need other approaches. And next slide, please. So those can be direct, which is what's being done in the lab today when you send your blood card in or your blood sample. That's by HPLC or mass spec. It might say amino acid analyzer. It's using those methods to measure directly and quantitatively the amount of phenylalanine in typically plasma derived from your blood sample. And there are other indirect methods that have been developed over time, and a number are very, very good of taking advantage of the chemistry of phenylalanine to give a detectable change, maybe color, fluorescence, changes in pH, and things like this. So these are kind of the main categories, really, of approaches that we can take. On the next slide is really our thinking about this, which is DNA aptamers. So the idea of DNA aptamers is that they bind a bit like an amino acid, but they're small, so they can detect small molecules efficiently. So it's short strands of DNA. The advantage of DNA, as you may be familiar with from all the detective shows, is DNA sticks to everything, right? So they always find it at the crime scene. It's very sticky. So the good news is it does stick to phenylalanine. And the trick is to make it stick with high specificity and sensitivity, so we know that we're really only measuring phenylalanine. So that's the kind of the challenge of development. The advantage is it's easy to manufacture and it's adaptable to readouts that it can be colorimetric or enzymatic or really electronic and things like that. On the next slide. So what the team at Columbia developed was really two adaptations of that aptamer to make it work for our Home Phe monitor. One is a short DNA strand that is released, that yellow quencher that you see. So that little strand, when the aptamer binds phenylalanine in the middle there, the circle, it will release that, which allows it to trigger a detectable event. And in the case of the assay we're using now, that's a fluorescent tag. So you got one fluorescent tag for every binding event, so one-to-one relationship between phenylalanine and the signal. The other thing is, you see that little red shell around the phenylalanine? That's adding a metal ion salt, which what it does is it essentially bulks up the phenylalanine so it's more stably detectable and makes the test more sensitive in a mixture of whole blood. So the idea here is that you can detect and measure the phenylalanine in whole blood, again, to make that assay simple, as I was talking about, and minimize the number of processing steps, ideally. On the next slide. So just kind of show you how that works. These are bland Altman plots of low range detection, 50 to 100 micromolar. And what we're comparing here is basically looking for the cluster of points around that zero line, the blue line in the middle, meaning these two methods that you're comparing are performing equivalently across a range of concentrations. Here you see at the bottom 50, 100, 150. Those are the micromolar concentrations of the phenylalanine. And those orange dotted lines are really the range in which it's acceptable to say those results are giving you equivalent data. Now, in a clinical test, you never have absolute point-to-point comparison from different methods just because of the vagaries of clinical chemistry. So this is how they compare, in this case, HPLC and mass spectrometry on the left. So those are the gold standards. And we want to see that same sort of clustering when we compare our aptamer to mass spec or HPLC on the right. And indeed we do. So that means our chemistry is giving us clinically equivalent results that we can then move through to give a home monitor test that would give us the same results as the clinical lab test. The next slide shows you the system as we envision it. So there's this simple disposable device. The cap has a little cup on it that you see sort of sticking out of the blue cap that collects 5 microliters of blood from a finger stick. Screw that into the device. The reagents are in a little pellet in there. So all that chemistry I showed you is occurring in that buffer, and that's where the actual reaction occurs. That takes a couple of minutes to react. And on the next slide, it shows you the kind of the system. So there's a reader, which is battery powered, Bluetooth enabled, and Zigbee capable. So it stores all the information. It does the analysis to convert that fluorescent signal into a number that you're familiar with, and connects to the cell phone app, which I'll show you in a second, that overall manages the test itself. So it gives you a complete system, sits on your countertop. The battery lasts a week. It stores a year's worth of results. So it's a nice, simple system. Has a lot of data checks. It reads a barcode on the disposable, so you know it's your test. You haven't used it before. It's securely linked so that it's your reader, not somebody else's reader, and that sort of thing. So it gives you the self-management, real-time data. The payers want, of course, a lower cost test and a more home use so that the patient doesn't have to come in. I'm sure you guys don't want to do that. More doctor's visits is really a burden on many families when they're not clinically necessary. They're just monitoring. And the pharmaceutical companies love the idea of clinical trial support and marketing support and compliance for new drugs that are coming on the market. Next slide. So this is just really quickly the software. So it's HIPAA FDA compliant, meaning it's secure, your login and everything going on. You see the data and the trend analysis. You can export it to other apps you might want to use for diet control or management, and of course share it with your physician. On the next slide. So so that showed you, sorry, that showed you the software running there. If you click it one more time, that's fine. And then the whole system is shown here, which is the chemistry with the reader and the cell phone app, and giving you kind of the results that we hope for across a spectrum of ranges of concentration, really from low range to what we might see for some adult patients as well, or patients that aren't under some of the new therapies. And on the next slide. So we envision this as a home system where it's basically providing you not just that real-time support, but ultimately for the pharmaceutical developers, clinical trial support, things like this, reimbursement support on the insurance side, and then for you, appointment reminders, prescription refills, connecting to your smart home systems as well. And on the next slide. So we're developing phenylalanine first. We have some other targets that we're working on through work being done at Columbia. We just started looking at MSU, for MSUD at leucine as well, which we have on feasibility, which we hope to bring into the test going into the fall. And on the next slide, which is just to say very quickly, we're Columbia spin-out focused on this area. A lot of interesting data that can be extracted from the system using the DNA aptamers and this smart home hub. So we really look forward to engaging with you guys as we bring this system online. Next slide. And with that, I'll say thank you and look forward to your questions at the end.
Mike, thank you for a spectacularly well-timed presentation. And again, as Mike said, if we can hold questions to the end, that will allow us to move through all the presentations, and we'll achieve that using the chat function. Now I'd like to introduce Peter Warto of Egoo Health. And Peter, welcome to the NPKUA Home Phe Monitor Webinar. Thank you for your time to present. I know you're in a vastly different time zone than us, so I thank you for that. And please begin.
Okay, can you hear me?
Yes, we can hear you.
Okay, yeah, I represent the European part of this competition, and welcome here from Copenhagen, Denmark. I represent a small company called QLife, and we have a home monitoring device called Egoo. So our brand is called Egoo Health, but the company is called QLife. I'm the CSO of this company. We were founded in Denmark 2 years ago, and we actually went public some months ago in March. We are currently 21 employees, and our product is a small medical device that can measure biomarker at home. Not only phenylalanine, but also other biomarkers. I will come back to that. Of course, we are following requirements from the EU for CE mark, both for professional use, but later also for home use. And the whole idea is that we need the same test done, and we just want it faster at your home instead of at the hospital. Our focus area is the PKU. That's our first priority, but we have also been involved in COVID-19 testing, both molecular and antibody tests. Next slide. So what is it? It's a small device. It's the size of a teacup. We call it Egoo. It's a device. It's a small capsule, and each capsule has a particular biomarker. It could be Phe, it could be CRP, it could be COVID-19. There's different biomarkers. Each capsule represents one biomarker. Then we have an app. So from your smartphone or your iPhone, where you can control the whole instrument. And then we have a cloud, where all the data is uploaded, and you can actually also have your physicians following your data. So if you have a particular doctor that wants to follow your measurements at home, he can actually log in from the hospital and actually follow the different results. But let's show a small video of the system so you get an idea of how it works. So this is the Egoo device, and here you see in capsules. And what you do is that first you put 10 microliters of blood into a small blood transfer pipette, and then you add the blood into the capsule like this. And after that, you insert you actually activate it by the iPhone. Then the iPhone contact first the cloud, and then the cloud contact the Egoo device, and the drawer opens. You put in your capsules in the Egoo, and you scan with your telephone a barcode that tells the Egoo I'm a Phe capsule, and here's my calibration data. And then the Egoo runs the analysis. If it's a Phe, it runs a Phe cycle. If it's a CRP, it runs a CRP cycle. It really depends on which biomarker you're talking about. And then after a certain amount of time, the Egoo is actually transferring the raw data to the cloud, and here you are calculating the result, and you go back to your smartphone, and here you can see the results. So that's just how it works. If you go next slide, so it's a blood, 10 microliters of blood. The test runs in 5 hours. So for us, it's not a 10-minute test. It's actually a 5-hour test, because we need to do some blood extractions and some waiting for this particular assay to execute and to generate the fluorescence-based chemistry. So it's not a 10 minutes test. It will take 5 hours, our test. Next slide. Now, if you go to a little bit of timeline, I show you this scenario and this timeline in Norway for some months ago. That was just before COVID-19. And the idea was there that we should start actually the clinical trials together with the Function Wisconsin in Holland around March, and then we should follow this timeline with different home users and clinical studies and so on and so on. But that was before COVID-19. Then COVID-19 came. Next slide. And just to show you, we were actually involved in corona testing, because it turns out that we could actually test corona on our Egoo device. We have system that called Big Egoo, where we could test around 1,200 corona tests per week. And I'm just showing you this because it's not only bad for the Phe assay monitoring system, because we learned a lot of stuff here. And we learned a lot of usability stuff, a lot of chemistries and so on, and what's good with our small Egoo and what needs to be improved and so on and so on. So even that we actually stopped in this period because we were doing corona testing, around 1,500 corona tests per week using our Egoo system, we actually learned a lot about our system and how it functions and where we should improve. And we have just started up our Egoo, our Phe development scheme again. If you go next slide. I just want to show you a little bit chemistry here. I'm just saying that in terms of testing the reagents, is working quite nice. It's not reagents that we have invented. It's something that has already been reported in the literature. Here I'm just showing you some calibration curve where you can see that there's a linearity between the concentration of Phe and the signal of fluorescence. And we have also done it with the Phe spike blood samples, and we see again very nice linearity. So from a reagent point of view, basic reagent point of view, it works quite fine. If you go next slide. That's just to show you where we are and what we have done. We have done the research behind the Phe assay. We have in the development, we have transferred the assay from the assay to the capsule, the Egoo capsules. And at the moment, we're actually doing a pre-study here in Denmark. And this pre-study is just a study where we actually are putting 7 Danish patients are getting a capsule each. And the idea is simple, that they should fill this capsule with 10 microliters of blood, and then they should send the capsule back to us, and we will analyze the results. And the reason we're doing that is simply because we need to find out, is it possible to actually add the blood into the capsule? Can you do it? And when we get the capsule back, we disassemble them and look for how they actually transfer the blood from the finger into the capsule. Does it look nice? Is it okay? And then we are comparing results with the results from the hospital. That's kind of, you can say, a pre-study, and that's what we are doing right now. Next is that in around 2 weeks from now, we will receive 200 samples from Professor van Swansen in Holland, and we will do the comparison studies where we compare with the standard equipment in his lab. That's very important. That's our first comparison study on the complete system. And this will tell something about the accuracy of the system. So we're really looking forward to that study. And you can say after that study, the next thing we're doing is that we are doing the rest of the assay for the professional CE mark, reproducibility, stability, interference, stability, and all this stuff that has to be done before you can have CE mark for professional use. And when we have the CE mark for professional use, we can start the feasibility study, which really involves real people having a home monitoring system, Egoo home monitor system at home. If you go next slide. Can I have next slide? Yeah. This is just to show you, this is the same timeline again. And what I just want to say that this is where we begin, and just 1 year delayed. And this slide is just telling you that you have seen that slide before, but you should just push 1 year. So you can say after the feasibility study, after the CE mark, the professional use and the comparison, we will start doing the professional protocols with the van Swansen and send Phe monitoring devices to home use to perform the feasibility study, because without that feasibility study, we cannot get the CE mark for home use. So you can say after we have the CE mark for professional use that I just showed you in the previous slides, then our clinical study can actually start with 50 patients. And the whole thing is that we will be 1 year delayed because of the corona, but it's not all bad because we have learned a lot of our ecosystem and where we should improve both from a mechanical but also from software point of view. So we are in good shape, and this is absolutely our first priority. So we are looking forward to let you follow our progress. So each time we will have new results, I will contact the society in US and enlightening them in those results. I think that's more or less from my side here in Copenhagen.
Okay, Peter, thank you very much again for another spectacularly well-timed presentation. Yeah, I think it's worth just reinforcing that that the idea that the COVID-19 pandemic has impacted research, particularly as it relates to PKU, for everybody around the world. And, and we are, research in terms of medical devices, in terms of therapeutics, in terms of gene therapies, have all been unfortunately delayed. But I thank everyone here, particularly for their diligence of trying to get things done in the context of a pandemic. So for next, I'd like to bring up Shima Dalarirad and Robert Harper from In Vitro Diagnostic Solutions to talk about the progress of the development of their Home Phe monitor.
Thanks, Lex. Getting a lot of echo. Uh-oh. IVDS. Well, Lex getting a lot of echo. Is you comfortable with that?
Well, I'm going to mute everybody, and I'll try to unmute you. And then you'll have to let me know, and Shima's going to talk.
Hello? Can you hear me?
Yes, go ahead.
Can you hear me?
Yeah,
that's better. Thank you.
Yep.
So In Vitro Diagnostics Solutions develops point-of-care devices for the diagnosis and monitoring of rare metabolic disorders. The presentation will be by myself, Robert Harper, President and CEO of IVDS, and Shima.
Hello, this is Shima Dalarirad. I'm the PKU lead scientist at In Vitro Diagnostic Solutions, and Rob is going through the slides. Thank you. Next slide, please.
Next slide, please. Thank you. So just an overview of the presentation. The slides 3 through 5 will demonstrate or describe the technology. 6 through 8 will look into the results for spiked whole blood samples. At this point, we weren't able to obtain actual patient samples, so we purchased whole blood and spiked it through the analytical range. Slides 9 and 10 are the regulatory pathway. Slide 11 is the timelines. 12 is our funding requirements to fulfill the PKU platform. And finally, slide 13 and 14 is our commercialization path, and then an overall summary. Next slide, please. Slide 3 is really the design of the test strip. It is a vertical platform. Blood is applied to the spreading layer, and the cells are filtered out through the primary and secondary blood membrane prior to it reaching the reagent layer, the fourth layer. The fourth layer contains an indicator or a dye that turns blue. The color intensity of the dye is read by a handheld meter. The color intensity of this dye on the reagent layer is proportional to the phenylalanine blood concentration. Next slide, please. This is the PKU Now test kit. It comes with a vial containing 20 capillary tubes for the blood draw, another vial containing 20 test strips with a hands-free capillary stand, optional 2 levels of controls, mainly for CLIA, and finally the handheld meter, which has connectivity. The meter reads the color intensity of that reagent layer and converts it to Phe concentration in mg per dl and micromolar in the output. This is all done through an integrated curve set. Next slide, please. This is really the directions of use for our home monitoring to show you the ease of use. You unpack the kit, you initiate the meter, you remove a EDTA capillary tube and place it in the capillary tube holder. Wash your hands, you do a finger prick and apply the blood to the capillary. It's a fixed 20 microliter volume. Bandage your finger, Then using that capillary, you apply the test strip, you close the cover, and then within 5 minutes you'll have a Phe result in micromolar and mg per dl. Next slide, please. So IVDS has received a phase 1 award, and then we did a phase 2 award several months ago, and this is some of the data that was in the phase 2 award. Excuse me. The x-axis on the left plot describes the results from the amino acid analysis using spiked blood samples, 1 through 25 mg per dl. The y-axis is the data from the PKU now. The plot reveals excellent agreement with an R² of 0.99. To the right is the Bland-Altman plot, revealing that there's only 2 points outside of plus or minus 2 mg sigma from the mean of 60 determinations. And the Bland-Altman plot really describes the agreement between 2 quantitative measurements. We did meet the criteria of less than 90 or greater than 95% of our data points within 2 standard deviations means. Only 2 points were outside of the 2 sigma. Next slide, please. So slide 7 is a plot of 3 samples, 1 which is a native sample, unspiked, 3 and 6 mg per dl phenylalanine spike. All of these samples were also spiked with 3 mg per dl tyrosine, which is really the upper endogenous concentration. And tyrosine has a very similar structure to phenylalanine, as it's recognized by the enzyme phenylalanine dehydrogenase. The elimination of tyrosine has really been the pitfall of previous development efforts. As you can see, the low concentrations at 1 and 3 mg per dl actually almost double for a false reading of phenylalanine or an over-recover of phenylalanine. And this is quite concerning when trying to maintain a balance of normal from 1 to 6 mg per dl, especially for like women who are pregnant and well-maintained patients. Next slide, please. So the CLIA is going to require, CLIA waiver will require, which is the clinical laboratory improvement amendments, will require two levels of controls. We've prepared synthetic controls at 5 and 14 mg per dl. It's very simple to use, and what the controls will do, placed in dropper bottles, will provide results to ensure that the test strips and the meter are working properly. And at both levels, we've seen excellent agreement, excellent precision and accuracy. Next slide, please. So in 2021, we anticipate having two preclinical sites with 40 PKU patients per site, not including control patients without PKU. One of the sites is under CHOP, Children's Hospital of Pittsburgh, under Dr. Vockley's group, and Jen Baker as well there, as well as another site at Cooper Medical Health under Dr. Egerton's supervision. This is going to be a self-testing protocol. What we're trying to do is to make sure that it's very simple to use for each end user prior to design lock of the PKU platform. The Children's Hospital of Pittsburgh will have the IRB oversight of the 2 sites. Next slide, please. So this is our regulatory team. I've known Claudia and Marcia for a long time. Claudia is excellent. She's going to be and has already started the implementation of our quality management system. This will allow us to manufacture in-house and assemble in-house. And Dr. Zucker is well known in the regulatory affair arena. She has over 25 years experience and has many point-of-care diagnostics gotten through the FDA. Next slide, please. So this is the projected timeline. As everyone else, the COVID's kind of set us back a little bit. We closed the shop for a month, and then we alternated each scientist each day. So we're a little bit behind, but we're moving forward. So we want to We're hoping to submit the pre-IDE to the FDA in 2020, as well as continue to implement our quality management system. We want to perform the preclinical evaluation and 3 clinical sites in 2021, as well as the 510K submission. I realize it's an ambitious goal, but that's something that we'd like to strive for. And then in year 5 is market launch. Again, we tend to want to manufacture in-house. We have a coating system, a slitting system, and assembly. We're currently negotiating a price point for our portable handheld meter. We anticipate the cost of the test strip to be $5, and we're looking for the end user to purchase the meter for about $300. Next slide, please. So on the right side, we believe this is the estimated funding that we will need for completing the task for 3 sites of clinical trials. We have $150,000. We believe we need $200,000 for the design history file, risk management, CE mark, and 510K approval, and then $100,000 for marketing and demonstration, disseminating the product, as well as surveillance. Most of our data was in the phase 1 application. It was granted by the National Institute of Health. That was for $225,000. We submitted a phase 2 application for $2 million, and it says pending review. We got a call or an email last night that the award was granted, so we're very happy about that. We would like to thank the National Institute of Health for their support, as well as the National PKU Alliance. So in summary, we believe our device is simple to use, it's accurate and precise, and a key feature, it eliminates tyrosine interference on phenylalanine dehydrogenase, the enzyme. It's designed to meet all the NPKUA specifications, and again, we'd like to thank the NIH as well as the PKU, National PKU Alliance. And that's all I have, so I'm not sure where we're on the time, but
that's quite a bit you have there, Rob. I'd like to thank Rob and Shima for the presentation. I'd also like to congratulate you guys on your phase 2 SBIR. That notification could not have been more timely for today.
I know, exactly.
So that's awesome to hear.
And just to that point, Lex, is that we had written in the funding for the preclinical sites. So the NIH will pay for preclinical, and unless we write another application for clinical sites, which we may, we will need to outsource that funding.
Okay. Well, that's terrific. So what I'd like to do, first thank all our presenters for excellent presentations and updates, and I'd like to open open up for questions. I think what I'd like to do, since we have 3 presenters and we can't actually see each other, just as a matter of making it happen in an understandable way, I'll ask the questions, and then we'll just have each presenter answer in the order that they presented. So I'll ask the question, Michael, you'll address, Peter, you'll address, Rob and Shima, then you'll address. And I think, we'd like to keep the questions, the answers to the questions as direct as possible. So the first question that I'm seeing is, what is the projected timeline for these devices to be available? Mike?
Sure, thanks, Lex. As you heard from a number of the presentations, COVID has, of course, affected timelines a bit. Our projection, kind of similar to the other teams, is pushing things out. We are have already negotiated our clinical trial design with the FDA, so that's good news. But we expect to be able to start that sometime in the fourth quarter of this year. And once that's completed, we would target launching kind of the first quarter of next year is the hope. And we certainly will, keep people apprised. But I think that's a realistic timeline right now.
Terrific. Peter?
Yeah, I mean, we have a lot of experience with the COVID-19, where we have placed some instruments and we have done some in-house analysis. But I'm not so optimistic as the previous, because here in Europe we need to have a CE mark from professional use first. So before we actually can do a clinical trial, we need to show that our data, our sensitivity comparison, so on, is perfect, and we can get the CE mark professional use. And then when we have the CE mark professional use, that means a doctor can do it, and all professional people can do it. But then we need to do the feasibility study, and the feasibility study is really finding out what can normal people, ordinary people, can they also do it, because that's quite important. That's why we have cheated a little bit and put some capsules out to people already now to find out, is it possible to get the blood in, do they get the same result, and so on. But I would say the short answer is, I think it will take approximately 1 year before we can actually complete the study, the feasibility study, and that means we can sell for home use. So it takes time here in Europe, and you need that CE mark for professional use first.
Thank you, Peter. Rob? Again, we're thinking about timing to market launch.
Yeah, it's a great question, and we're proceeding as fast as we can. The path to market is always more difficult, to be honest, than the actual chemistry. I mean, we're looking, for us would be year 5 in 2022, and that may leak into 2023. But that being said, our first target market is going to be through the clinics, right? So we want the physicians and the caretakers to have a real understanding of the device and the accuracy and performance, and then train those patients to use it at home. So our dissemination pathway is through the PKU clinics. There's a learning curve there, and then once they have a really good understanding of the performance and the the procedure and directions of use, then they can train the patients to take it at home. And I would imagine it's going to be a prescription to take those devices and test strips home. It's much easier, I believe, that way as a market path than it is for the FDA to clear right to at-home use.
Okay. The next question, I think all of you could easily predict, and that is, what are your guys' projected costs to the patient at the time of launch, and do you see those prices changing with time? Michael?
Sure. Definitely the costs change over time because there's volume opportunities, right? The more testing that's done, the lower the cost of manufacturing. I think the reader cost to the patients is all going to be probably pretty similar for everybody. I don't know, but for us, we're estimating it's in the few $100 range. I think for the disposable strips, we've had some discussions with insurance companies around that, and it's honestly hard to predict. Our target is that our cost of goods is low enough that however they measure the user price depends a little bit on your insurance, et cetera, and how it's reimbursed and how many tests they allow you to have per unit time. But the goal is to make it at least a third lower cost than patients are paying today, with the goal that they're able then to do a reasonable increased amount of testing, which patients want and clinicians seem to want as well. Sorry, I'm not giving an exact number because I don't know yet, to be honest, until we're closer to the clinical trial. But we've set those objectives to make it useful to the patient, useful to the insurance companies, and meeting the clinical targets of increased testing.
Okay, thanks, Mike. Peter?
Yeah, what I can say.
I know we might have to do a currency exchange rate here.
Okay, but I mean, to very short, the Egoo is run by either iPhone or a smartphone, and it costs the same as an iPhone. Okay. And in terms of the capsules, it will be around $10 per capsule, and it really depends on the here in Denmark there will be reimbursements. I don't know your system in US, but it's around $10 and an iPhone.
Okay, perfect. Thank you. Rob?
We've had so many different, so much difference between state to state, insurance, what's reimbursed, what's not, the pricing of reimbursement, how it's going to be reimbursed. So I could tell you what our cost of goods are. Again, we're negotiating the meter price, but we're still trying to figure out how the reimbursement's going to occur and how that impacts us. We're actually looking at a kind of a closed loop ecosystem where we provide each patient 4 strips per month with the meter, and as well as consultation from the nearest clinic. We bundle all that into a package. That's going to be our approach. But as far as the meters, we believe will be around $300. The test strip will be around $5 per test strip. But I can't tell you. I've really struggled to figure out what the reimbursement for that is and how that's going to take place.
Thank you. Yeah, I think it's that is a very complicated question right now, because obviously as you manufacture more of anything, any new product on the market, the cost, the manufacturing costs can be driven down over time as the number of units manufactured go up. And then we have the added complication of reimbursement from insurance companies, and that's a complicated process and takes some time too. So I'm sympathetic to our presenters on the difficulty of giving just a straight up number, because there are many factors that we're still not aware of right now to give that. The next question, I think that actually, Michael, this was directed at Aptatek, but I think it might apply to all of them. And the question simply is this: how many patients do you anticipate that you need for your clinical trials to apply for marketing approval with the FDA? Michael?
Sure. Thanks, Lex. That's a great question. The design that we've discussed with the FDA is to demonstrate that the system gives the same results, equivalent results to the satisfaction of the FDA as the current tests that the patients are using. In this case, we will benchmark against mass spec. We're anticipating a total number of patients is in the 35 range, plus 5 pregnant women as well, because they have some special requirements for testing. And they will be in ranges of age, so juvenile to adults. Just by availability of patients, we expect that to skew towards young adults to some degree, but we're hoping to get a wide range of patient ages in that study.
Terrific. So sort of 35 to 40 patients. Yes. Total?
In total, yeah.
Yeah. Peter, have you all thought through your clinical design process enough to know how many patients you expect to be required for approval?
Yes, absolutely. We are in the middle of filing our approval with the Dutch authorities because we are running our clinical trials through Van Sponsen in Holland. But you know, we anticipated 50 PKU users, but we actually are finding out that 25 is actually enough. But you have to remember here in Europe that we need the CE mark for professional use first. I know I've mentioned that before, but we cannot. I mean, if you don't have the CE mark in Europe, you don't really. The authority asking, how can we even be sure that your instruments are working properly? So they want to see a full data package for the CE mark for professional use before they allow a clinical study in Europe. Otherwise, they say it's a no-go. So we need to do that first, as I also show you on the timeline, and then after we have this one, we will start the clinical trials with the Franklin in Holland, and but 25 users, that should be enough. And also, there will be many small clinical trials. First, for example, we are cheating a little bit in the moment and actually sending out our capsule to 7 users just to ask small questions that are extremely important. Can you put blood in? How are you operating the system? It's always, you know, when you are in development and you see how people actually are doing, they say, no, that was not the intention of doing that, and we have to go and, you know, reconstruct something and so on. So there's a lot of small clinical trials going on, but the big clinical trials will be after the CE mark for professional use next year.
Rob?
I'm sorry, can you repeat that question?
Sure. Yeah, the question was, how many patients are you anticipating for a clinical trial, for a definitive clinical trial to get approval?
Yeah, I mean, we have a donor study protocol written, and that's been sent to Children's Hospital of Pittsburgh for IRB review. We're looking from greater than 3 years old, up to late adult, who may actually need some help with, the finger stick and the sample application. And I believe we're looking at 50 per site for the clinical trials. There's a certain statistical criteria which my statistician can tell me, which is over my head, so I would refer to him at this point, but I think it came in around 50 per site to be significant.
Terrific. So another question that I've received a couple of times now is, that once the clinical trials are initiated, how would people find out about the clinical trial and, sort of apply, if you will, to participate in the clinical trial?
Thanks, Lex. So the trials are in the U.S. are registered in the government clinical trials website. So it would show up there once it's officially submitted and enrolling patients. And kind of like the other teams, given the size of the trials, we're focused on a couple of clinical centers, one in Pittsburgh also, and one in Boston. And so we'll make people aware of that as we get that registered. It's to certainly to recruit patients who are in those communities, since that would be a great help.
Great, thanks. Peter, I know this is kind of a harder question for you since you're located in Denmark. But should we have any patients that that work through the NPKUA, how would and happen to be in Denmark, how would they find out about the clinical trial? Maybe that's the best question.
Yeah, I mean, here in Denmark, we have a lot of questions through our website. I think we have more than 30 or 35 people that wants to do this trial in Denmark. So we don't have any issues with recruiting people here in Denmark. And But our clinical trials is the big one, the final one will be through the Professor von Sponsen in Holland.
Right.
And so he will be the one who's recruiting the patient for that trial. So that's how it works in Europe. But you know, we have discussed before also with US if there could be some collaboration there, and we could place some devices. But I would say, you know, our first priority is in Europe and Denmark, where we are learning a lot of how our instrument works and what can be improved and so on. And we are getting there. But I think we should talk, and I know we have already talked, and we should talk some more on finding out would it be possible to place instrument in US. But that will be something that has to be taken care of your society, or we have to find some kind of collaboration together, because here right now we are 100% focused on Europe and Denmark, but we are very, very open to that question.
Terrific. Rob?
Yeah, I was actually hoping for the NPKUA to help us out with on your site. Yeah, there's a lot of East Coast clinics, but I've also been corresponding when I was at the NPKUA in Atlanta, and I've met with one of the presenters there, as well as his grandchildren both have PKU, and his daughter was asking me, is it possible to get a clinic in Alabama? And I think that would be interesting to have a site that's, they have to travel a long way to their site, and
right.
It'd be nice to have a kind of a different subset there. So, you know, we're looking at Cooper Medical, which is walking distance from me, and Children's Hospital Pennsylvania's and Jen Baker has been excellent. So yeah, we're looking at a third site, and I'm trying to reach back out to one of the presenters. He was at Clemson. He was developing a like a post-it or with creatinine, a urine creatinine post-it. I don't know if you recall that or not, but
I do. We can. So you know, if we want to reach out later, we can help make that connection. To the bigger question, that yes, the National PKU Alliance operates what we call a patient registry, and the patient. One of the questions that we have in the patient registry is, are the new registrants, the PKU patients that are registering for the first time, are they interested in participating in clinical trials? And do you, as the patient, give the NPKUA permission to help make connections between patients and clinical trials? So that's one very efficient way that we can work with companies developing either medical diagnostics, medical devices, and of course therapeutics. There's one more question on here. I think I'm gonna go ahead and answer this question in the interest of time here. Was that there was a question, will the studies in the US provide information needed for CE marketing requirements in the United States? And so as we've heard from Peter, that there's a European equivalent of the FDA, and the FDA controls and regulates, in this case, medical devices approval in the United States and Europe, and the EU agency does the reverse. They regulate the control and approval of devices in the European communities. But there is quite a bit of overlap. And typically what happens in these scenarios is that once a device or medication, for example, is approved either in Europe or in the United States, there is a mechanism where you can use some of your existing data. Let's say if you were an American company applying in the United States, you can use some of your existing data in the US as part of your application in Europe, but you also have to do some clinical trials in Europe and vice versa, going the other direction. But the data from one company, as I'm trying to read this question here, the data from one company will not generally be applicable to another company's effort to develop their product. So those would have to be handled on a product by product basis to carry out those bridging studies to allow the transfer of marketing from one community to the other. So with that, I would like to, and sadly have to bring this to an end because I think the presentations have been excellent. The discussion has been excellent. Participation from the viewers in terms of questions submitted has been excellent. But I'd like to thank everybody for participating. I'd like to thank everybody for those who presented for presenting, and I'm excited personally to see that we're rapidly approaching the finish line to get this important medical device out into the community. So with that, Michelle, I think we'll bring this to a conclusion, and if anybody has any additional questions, I would encourage you to, one, you can reach out to the NPKUA and we can help provide information. Two, you can visit the company's websites, Aptatek, Egoo, and In Vitro Diagnostics, to keep track of their progress. And three, in the United States, you can watch the website clinicaltrials.gov, which is the official government-operated registration site for all clinical trials going on in the United States. And you can be on the lookout there for the initiation and enrollment for Home Phe Monitor clinical trials. So again, thank you very much and for participating, and everybody have a great and COVID safe day.